Now let's turn our attention to the transport of sugars through the phloem vessels of the plant. Sugar is going to be produced in photosynthetic organs, namely the leaves of the plant, although some stems in certain green plants are also capable of photosynthesis. The sugars produced in these green leaves and stems is going to be transported into the phloem. Here we have these sieve tube elements, and it's going to become phloem sap. Phloem sap, by definition, is going to be an aqueous solution, meaning water is a solvent, and it is going to be high in sucrose. We've learned in biology that photosynthesis leads to the production of glucose, and some glucose molecules will be modified and eventually lead to the formation of sucrose, which is a sweeter type of sugar, and that can be loaded into the phloem sap. The places where photosynthesis takes place are going to be known as sugar sources. Here again, think about those structures, the green parts of the plant I just made reference to, such as the leaves and the stems. We call them sugar sources because that is going to be the net where the net production of sugar will be taking place. Phloem will be transporting sugars from sources to sinks. The word sink here refers to organs in a plant, like maybe a modified stem or a modified root, where carbohydrates such as sugars are going to be stored. How does that transport happen from sugar sources to sinks? Let's take a look. Here in this illustration, we have an arrangement of cells like the ones you can find at the vascular bundles of a leaf, also known as veins. In this illustration, we see, for example, cells of the mesophyll. Mesophyll cells are going to be the ones responsible for photosynthesis. But before these mesophyll cells move their sugars and the sugary solution over to the phloem, those sugars must pass through the bundle sheath cells. These are cells that are surrounding and providing protection to the veins of the leaf. From the bundle sheath cells, the sugary solution is going to be moving over to certain cells of the parenchyma. Now we're inside of the vein. Now we're inside of the vascular bundle of the leaf. And here some water and sugars may be moving through the apoplast or through the symplast, but all is going to be collecting into these sieve tube elements of the phloem. In order to load sugars in high concentration into the sieve tube elements, there's going to be a need for active transport. And the specific type of active transport we're seeing here is something called co-transport. Let's explore this for a moment so we can understand how the sugary solution of the phloem can be so rich and so concentrated in sugars. First, a cell would have to use ATP to create a high concentration gradient of hydrogen ions. Look at this illustration and see how hydrogen ions are going from a region of a low hydrogen ion concentration to a region of a high hydrogen ion concentration. Once this high concentration of hydrogen ions has been achieved, a different transport protein would allow for hydrogen ions to cascade back into the original compartment from high concentration down to low concentration. At the same time, this special transport protein that is allowing hydrogen ions to return back to a low concentration will also allow for sugars to be moving from low sugar concentration into a high sugar concentration. And so notice that hydrogen ions are going down their concentration gradient, but sugars are going up against their concentration gradient. Because for all of this to happen, energy was required to begin with. And because initially hydrogen ions were going to a high concentration, and because sugars are also going to a high concentration, this type of transport is going to be known as active transport. So here we're going to have cells of the phloem that are going to be aided by companion cells. And so these companion cells are going to be largely responsible for this type of work that we have just described, the work of the plasma membrane doing the co-transports. We can create this rich sugary solution inside of the phloem of the plant. Now, sugars can go from the source, which is going to be the leaves, down to the sinks. Another detail that I wanted to point out to you is that when all of these sugars are being loaded into the sieve tube elements of the phloem, at the same time that's happening, water will also be moving into the sieve tube elements. And we can see that better in this illustration that I have. Let's take a look at what happens here as we look at the cells of the mesophyll. So this is going to be a photosynthetic cell. Eventually, these cells will be moving their sugar by active transport from low sugar concentration into a higher sugar concentration in the sieve tubes of the phloem. This is going to be followed by a movement of water. Water will always go from where you have a greater water concentration to a lower water concentration. Or, as we've learned in this chapter, from greater water potential to a lower water potential. You may ask, how can I say that there is a lower water potential on this side? The answer is because of all of these sugar molecules. All those solutes are going to be binding to water, attracting to water, and so that makes that solute potential become even more and more negative. Consequently, here we have a lower water potential, and that is always where water will want to go, where you have the lower water potential. Now that this is happening, think for a moment. This is a limited amount of space. This cell we call the sieve tube element. There's only a certain space, and there's all this sugar coming in, and there's all this water coming in. This is going to now develop a positive pressure. And the only way this solution can go is going to be away from the source down towards the sink. For this reason, we say that bulk transport in the phloem is going to be driven by positive pressure. This is also known as pressure flow, and it's characteristic of how sugars are transported from sources down to sinks. One of the things that may happen to the plant is that there may be too many sinks, too many structures that are taking in the sugar, and maybe not enough photosynthesis is taking place. What some plants can do is that they can do some self-thinning. They may actually voluntarily release, like shed leaves or excess fruits, which can also be a sink, and that is going to help them retain more of the sugars they actually need for their own metabolic purposes. Another name we have for the movement of sugars from the sources down to the sinks is going to be known as translocation, and by now you've learned that this is all going to be driven by positive pressure. Pressure that is created as sugars are loaded by active transport into the sieve tube elements and all the water that follows over here. And before I close, just to add to the pressure and how pressurized these sieve tube elements are going to be, you know that water is moving upwards from roots to leaves through vessel elements of the xylem. And because of the proximity of xylem vessels and sieve tube elements of the phloem, this high concentration of solutes, this sugar over here, will also end up drawing some water from the xylem over to the phloem, adding to the pressure that is going to help translocate these sugars from the source down to the sink. And I think that pretty much does it for the topic of transport of materials inside of plants.